Hi everyone. Thank you for joining the third webinar in our series, Use of New Approach Methodologies for the Risk Assessment of Pesticides. This series is being co-organized by PETA India, the Biological Agri Solutions Association of India, and the Society for Alternatives to Animal Experiments India. I'm Dr. Pandya and I'm an advisor to PETA India, and I'll be co-moderating today's webinar with Mr. Vipin Saini from the Biological Agri Solutions Association of India. Today, we will hear first from Ms. Hirna Pandya, United Phosphorus Limited, who will be presenting on non-animal testing methods for skin sensitization. Before I introduce a first speaker, I would like to point out that the information about the webinar series can be found at the link on this slide. There will be time for questions for the speakers at the end of the presentations. Everyone is on mute, but you can type your questions or comments in the question section within your GoToWebinar toolbar at any time during the presentations. This toolbar should be on the right-hand side of your screen. Please do mention which speaker the question is being addressed to. So speaking today first will be Ms. Hirna Pandya. Ms. Pandya completed her MSc in Zoology from MS University, Baroda, with specialization in wildlife biology, ornithology, and biodiversity. She has worked as a GLP study director for various short-term and long-term toxicity studies at different contract research organizations. With a strong experience of agrochemical regulatory and contract research organizations for almost 15 years, she currently works with United Phosphorus Limited as a global regulatory toxicologist. It is now my pleasure to turn the floor over to Ms. Pandya.
Hello, everyone. Good afternoon. And I would like to thank Ankita and all the PETA team members for inviting me in today's session as a speaker. And I'll not take much of the time, and we will begin the session. My today's session is for the overview on OECD methods for skin sensitization, and my session partner, Dr. Rahul, would be covering up on the case studies related to these OECD methods. Before we go into the details for OECD methods for skin sensitization, I would like to introduce a short base on what the NAM approach is and why we need them and how is it helping us in the pesticide regulatory risk assessment. Also, I would like to say that when we are going through the slides, we will have a more of an overview of the OECD methods and try to see the summary of which methods can be used for the testing. When we talk about the NAM approach, the first thing which comes into our mind is non-animal approach method, how it has been important, why we need it, because there are principles of 3R which are driving this concept of alternative testing. Alternative testing plays an important role in regulatory arena, and over the years, there has been a lot of work done and evolvement taken around alternative toxicity testing. These toxicity testing methods have a regulatory goal for classification, labeling, and transportation of chemicals and occupational safety. Not all in vitro methods are completely developed, but many are under progress, and along with OECD and other regulatory authorities and with the help of industries, there are a lot of consistent efforts going on in driving these methods to be developed. So when you see the slide of non-animal approach, it is nothing but a kind of a reductionist approach, which we can see that from the whole animal testing, or from the whole animal testing to a tissue and now to a body on a chip. So that is less is more. So with this non-animal method approach, we can do a lot of testing in a short period of time for many number of the chemicals by which we are surrounded in today's world. Now, when we talk about the NAM approach, another point which comes to mind is that why these NAM approaches are required. And we can see that there are multiple stakeholders which are driving these NAM approaches, like industry, CRO, trade associations, animal welfare groups, academia, regulatory bodies, and public together to make this 3R principle effectively working to develop the methods. These methods are ongoing and has an evolution on many levels to increase the productivity and harmonization of the methods take place for these. Now, talking about NAM approach, in this slide, you can see that there is a traditional approach which was there previously, and it is still under an existence. So we can see the two aspects on this slide about the traditional six-pack acute toxicity testing, which exists, and there is a modern in vitro toxicology perspective, which is also known as modern 21st century toxicity testing. Overall, we all have a common goal when we do these studies under a NAM approach or let's say traditional testing to achieve the toxicity endpoint and the classification. Here you can see the various toxicity categories which are listed out under various categories for understanding the classification. On the first session of this webinar series, we had Dr. Jain who had nicely explained about all the overview of these methods, and these are also gaining acceptance level in India as well, and that reference slides are also available on the website. Now we come to our main topic. What is skin sensitization? Skin sensitization is nothing but a type 4 delayed hypersensitivity reaction. A chemical substance, when it comes in contact to the skin, it causes an allergic skin response, and over a period of time, it develops into a skin sensitization. Now, we will look to a short video to understand that how these non-animal testing methods and skin sensitization is important to test and what is its importance.
Avoid animal testing.
I'm sure that you got a good glimpse about what we are talking in terms of skin sensitization and why it is important. Now we go to the first slide for the overview on the OECD methods which are available. Traditionally, first which came into existence was OECD 406 and 429, and they cover the local lymph node assay, Buller test method, and maximization test method. These all are in vivo methods, and all of them are helpful because they provide us an endpoint for classification, and some of the regulatory agencies still require in vivo testing. But we will see in coming slides what are the other alternatives available for the testing. So This slide shows about the adverse outcome pathway. We all know that skin sensitization is nothing but it is initiated by a covalent binding to the proteins. And once this starts, then the various key events and molecular events which are part of this skin sensitization reaction are taking place. Before we go ahead with this, I will have one more video for all of you to understand the two main important phases of skin sensitization reaction. Here we go. So I'm sure now you understand the two important phases which are part of a skin sensitization reaction. One is the induction phase and another is the elicitation phase, elicitation and induction phases. So when we talk about the NAM approach, it has evolved with the adverse outcome pathway, where there are various key events which take place at the cellular and molecular level, which are responsible for the reactions and adverse outcomes. And these all are testable events under various guidelines: OECD 442, C, D, E, and A and B, which is of course OECD 429. So each key event is covered under the guideline method to understand the adverse outcome pathways. Key event 1. The key event 1 is a molecular initiation, involves the covalent interaction of the allergens with the skin protein. The 442C method, which is conducted with the method known as the DPRA, that helps to evaluate the endpoint on the positive reaction. The second key event 2 is keratinous site activation, and that helps to release all the inflammatory signals and changes in the gene expression. We will not go much into the detail for each of the key events and the OECD methods because we have a limited time. OECD 442D covers the key event 2. Key event 3 is about the dendritic cell activation following an exposure to the antigen, which is covered under OECD 442E. And the various 442E methods are HCLAT, USENS, IL-8 look assay, and newly introduced genomic allergen rapid detection test, which was introduced last year by the OECD. The key event 4 is most important one, but right now there are no non-animal validated methods available for key event 4, and OECD 429 LLNE is one of the in vivo methods which is prevalent from quite some time in the industry for the testing. Genomic allergen rapid detection test is nothing but a simple test in which sensor cell is being used, which is of a human myeloid leukemia cell line, and the DV value which is derived helps you to predict the sensitization or non-sensitization of a chemical. Again, GUARD is of a key event 3 molecular event method, and it has an ability to detect the skin sensitization. Again, since we saw so many methods in our previous slide, I would like to comment that one size may not fit for all. There are so many in vitro methods available, and one single approach may not be helpful. To overcome this, OECD published a 497 guideline, which is a defined approach for the skin sensitization, wherein they have described about the in chemico, in vitro, and in vivo skin sensitization, which can be together used in various combinations for getting the endpoint of skin sensitization evaluated. EPA also had published an policy for replacement of the skin sensitization animal testing strategy. Skin sensitization, as we have spoken about the various key events, is not only one area, but it is also the correlation of the protein reactivity with the skin sensitizer. So when protein reactivity is one of the key events, it is not one standalone which will help you to recognize the sensitization endpoint. You will have to couple it with existing data in silico methods or the in vivo method. If there is no option available, then you go for in vivo. Otherwise, the in vitro methods can be used in combinations which were listed ahead. Here are the defined approaches that are acceptable under the regulatory submissions. That is the AOP 2 out of 3, means you can cover from key event 1 to 3, coupling two methods with the other one, either all three of them or either single method or three of them or two out of them to get the skin sensitization endpoint, which is the hazard only or potency for the chemical. Coming towards the keynotes and the advantages, why we need these in vitro methods, and the advantages are they reduce the reliance on animal testing, there are animal welfare benefits, they are cost efficient, they are quick, they can take some time, but then there are a lot of advantages, and important is that our endpoint of skin sensitization, is it a sensitizer or a non-sensitizer, can be derived. Apart from this, applicability domain is an important point when we are using these guideline methods. Each method comes with its own limitations for the type of the chemical which is getting tested, and we need to take care about those points which are explained in each of the guidelines. Some of the fiscal properties like KOP or KOW is very important when you are testing it with the OECD 442E method. Overall, we can say that NAM's various methods are available, and skin sensitization is one of the methods which has been really efficient tool, or you can say a combined tool along with the OECD and various methods available as an example, which has evolved over the time and helped us in understanding more in detail for the mechanistic events which are part of the skin sensitization. I will touch a small base upon the in silico models, which are also described in the OECD guideline. Those are the direct OECD QSAR toolbox and the QSARs for skin sensitization. There is a link which will help you in getting the information or the skin sensitization prediction at the first point when you have to test the chemicals. And that's why in silico model in combination with the other testing methods can be really helpful when you have to test your product. Here is another reference. ECHA has also very nicely published a document in October 2021, which details about all the in vitro methods and the endpoints derived and the combination which can be used for the chemicals. OECD guidance documents number 255 and 256 are also part of the integrated testing approach along with the refined approaches which can be used together for testing of the chemicals. And these are some guideline links for the reference for all the methods. As said earlier that we do not have one method to be tested. We have a several set of methods to be referred. So there are a lot of links over here for a reference which can help you to read through the various methods and use them during your testing and regulatory submission. So we come almost towards the end of our my presentation, and thank you for listening.
Thank you so much, Ms. Pandya. I'll request everybody, I mean, all the organizers and panelists to please put themselves on mute. So thank you so much, Ms. Pandya, for the wonderful presentation. It was quite insightful. I think the videos that you presented was, I think, quite helpful for people who are, trying to understand about this non-animal method. So yeah, so moving on, our next speaker today would be Dr. Rahul Date, and he would be presenting some case studies using these non-animal skin sensitization test methods for toxicity assessment of pesticides. Dr. Date completed his PhD in the field of biochemistry at the University of Bonn, where he established a novel role of the cannabinoid system in cutaneous hypersensitivity. He has worked for Sartorius Germany, Mars Germany, and Unilever Research India in different capacities and involved in method development for lentiviral isolations, antioxidant effects of natural products in tea biochemistry. He's also worked as a postdoctoral fellow at the University of Bonn, Würzburg, and Munich, where his prime focus was on cancer research. Currently, Dr. Date is working as a group leader at J Research Foundation, where he is leading a team of scientists who are focused on alternatives to animals using in vitro and in silico approaches. And his team is quite successful in developing assays like photosensitization and LCMS-based thyroid peroxidase activity measurements. His research findings have been published in journals of high repute like Free Radical Research, CR Talks, Altex, and Science. It is now my pleasure to turn the floor over to Dr. Date.
Thank you, Ankita and Hirna, for a nice introduction about the topic. Right now, we will be presenting in next 20 minutes the efforts put by the different groups, how to use different in vitro methods to predict skin sensitization. So, is my screen visible?
Yes.
Okay. So I'll be talking about skin sensitization in general. So as my earlier speaker here has mentioned nicely about what is allergic contact dermatitis, ACD. Basically, skin sensitization can occur in human by the repeated exposure of a particular chemical called a sensitizer. And it can be termed with different ways, like it can be called as ACD, it can be called as delayed hypersensitivity because it occurs after the second exposure or repeated exposure. And basically, what it differentiates this sensitization from the other types of sensitization is this is cell-mediated response. So it's a T cell mediated response. So it can be sometimes called as T cell mediated or chemical induced type 4 hypersensitivity. So many examples, like we know that poison ivy or the hair dye chemicals, what we use, PPD, paraphenyl diamine causes sensitization. These are the examples of ACDs. So if you look at this cutaneous hypersensitivity, basically it's as we have seen in earlier video, it's a mechanism where different serial chain or event occur in flow. This is one example where we have taken one simple compound called as 2,4-dinitrofluorobenzene as one example. And over here, if you see this small molecule when it's applied on the skin, it acts like a small haptane. So this penetrates inside the skin, and further this molecule is presented to the skin proteins. So once it's presented to different skin proteins, they react with that hapten, and further they the Langerhans cells or the dendritic cells what are present in our skin present these cells, present these molecules to the draining lymph node. And in the lymph node, as we have seen, it's presented these to the naive T cells, and further when the T cells get activated, then we have some sort of a specific chemical specific T cells which get activated, and these mature T cells, what we call it as, they are circulated inside your system with the efferent lymph system. And if you have, suppose, repeated exposure of this molecule, then you would end up into a sensitivity, or what you can see in the last stage of 7 and 8, where are different cells, different cytokines take a place role, and then you would find some sort of a allergic response elicited by these compounds. So this is a typical mechanism how the sensitization take place in our system. As we had seen, it's nicely
Dr. Date,
presented by.
just a sec. Dr. Date, I'm sorry to cut in between. Can you just present your slides in the presentation mode, because we're seeing a smaller screen of your current slide and then seeing your next slide separately. So there may be an option to show the slide in the presentation mode.
Just give me 1 minute. Still, you could see that?
Yes, I'm still seeing the present slide and the second, like the upcoming slide. Is there a way to fix this?
Ankita, still you see 2 slides, or how is it? Which mode?
I'm not able to see anything right now, Dr. Date. I mean, there's no slide visible on the screen.
I think from my side it shows it's showing. People can see your screen. Can you just check with the your technical support?
Chris, do you mind pulling up Dr. Date's slides if the same thing is happening at your end?
So doesn't it work if he clicks on slideshow from beginning on the left-hand side on the window that you just had? Go to the left where it says from beginning. Doesn't that work?
I think we're still seeing.
Okay, then I will pull up the presentation. So I will make myself the presenter and show the presentation. Okay.
Okay. Dr. Date, Chris will be presenting, and then you can just ask him to advance the slides as you keep moving forward.
Okay.
Okay.
Is it visible now?
No, so Chris would be presenting his screen, the slide on your slides on his screen, and then you can just ask him to advance the slides.
Okay, fine.
Okay, thank you. Thank you.
Do you see now the slide?
Yes, I can see now.
Okay, then I suggest we go to the slide where you were.
Okay, can you go to slide 4? Yeah, I think that's fine.
This one?
Can you go back, please? That's it. Okay. So fine. So as you could see in this slide, the complex phenomena of skin sensitization has been characterized into the key events what occur in sensitization. For example, a chemical has to penetrate through your skin, then it gets metabolized, and then, as I mentioned, the protein from the skin reacts with this chemical, and so you would have some sort of a covalent binding with skin protein. And the amount of the compound or the which reacts with your skin proteins would decide normally how the compound is sensitizer or not. So higher you have a reactivity of with your skin proteins, the tendency of this compound to be sensitizer is more. And this can be very nicely studied with the OECD guideline 442C by a HPLC based assay called as DPRA, which I will be explaining a bit in the next slides. So there are similarly Me-Too assays like ADRA assays, or the assays where you can also look at the kinetics called as KDPRA assay. They all lead with this key event 1. So if you go back to the next slide, to the next event called which is caused by the cellular response, the response by the keratinocytes. This can be studied with the OECD 442D guidelines, where the keratinocytes, when you have some sort of interaction with this compound, the NRF2, which is normally associated with CAPE1 protein, is translocated to the nucleus, and then further it elicits the response, and that can be easily seen with the luminescence, and one can predict the nature of a compound with this assay. The other assay which is used for mimicking the dendritic cell response, the key event 3, where you can use the THP1 cell line or the MUS cell line, and you could see the response of this chemical when you incubate these cells with these cells. So upon the exposure with this compound, you would have the expression of different receptors on these cell surfaces, and the receptor expression can be quantified with flow cytometer, and one can also predict how good the compound has a tendency to cause skin sensitization. The other response, key event 4, basically deals with the T cell proliferation. It's an in vivo experiment which Hirna has nicely explained, where you use different mice models to replace the what you call the classical old methods which were used earlier. And then nowadays we can also use different silico approaches, where we can use different softwares like Derek or OECD Toolbox, which are available. And how to use all of them is nicely explained by the guideline OECD 497. Can you go to the next slide, please? So as we have seen, sensitization is a very complex phenomenon. So none of these single assays or single key event measurement would help you to predict whether the compound as such is sensitizer or not. So what you have to do is you have to use a combinational approach, where you have to mix different assays in a serial ways or parallel ways, or you have to make some sort of a combination system where the output of this assay makes some sense. For example, for the first assay, DPRA assay is an in chemical assay where the metabolism is not present. It can predict up to, let's say, 80 to 85% whether the compound is a sensitizer or not. If you look at the second key event, the response from the keratinocytes, that is also an assay where it lacks lysine. The compounds, if the compound reacts with the lysine, you could not easily predict with the keratinocyte assay. But if you use combination of these two assays, you could get a sort of a 90 to 95% accuracy. So that's why a combination of such methods is very important, and there have to be a logical way where how to integrate these different approaches to make the meaningful conclusion. Can you go to next slide, please? So as I said, this is very nicely explained in this guideline OECD 497. What combination you should use, what logic it will make. Can you go to next slide, please? So one of the classical approach what we were taking till now was 2 out of 3 approach, where you take the output of 3 different assays. For example, DPRA assay, keratinocyte assay, HCLAT assay, which correspond to the different key events. And if 2 of them give some sort of a sensitization or non-sensitization output, we call that compound as a sensitizer or a non-sensitizer. Can you click, please? So this is explained in this publication. Next click, please. So the other two approaches called as ITS V1 defined approach or the ITS V2 defined approach. Basically, they deal with the outcome of two different assays which are explained in this key event assay, where you study the DPRA HCLAT output and combine that with the silico outputs. For example, in the ITS V1 approach, you have to use the output of Derek, or whether if you use V2 approach, then you have to integrate the output what you get from the OECD Toolbox 4.5. Can you go to the next slide, please? So this is explained in the OECD 497, which I will be covering in the next slide. Can you click, please? So first assay, the DPRA assay, is what is studied in the key event 1, where you look at the covalent binding to the skin proteins. Basically, what you do in this assay is you take two synthetic peptides, as shown over here, and incubate them with the test compound. After 24 hours, what you do is you look at the HPLC analysis. Can you click, please? And you look at the how much of the free peptide is present after this 24 hours incubation. The concept is, if the compound has a nature to react with these proteins which are present in skin, it will cause depletion in this peak area, and that can be easily visualized with HPLC instrument. And higher the reactivity, lower the peak you will get, and the compound have a more nature to react with your skin proteins. Can you go to next slide, please? One more click. And depending on how much of these peptides have been reduced, you can predict whether the compound is sensitizer or not. For example, if it causes more than 6.39% of the depletion, you can easily call it as sensitizer. The other key event 3 depicted by the HCLAT assay, where you look at the dendritic cell activation. So here what you do is you incubate your TSP1 cells, which is a surrogatory for the dendritic cells, with your test chemicals for 24 hours. And after 24 hours, you measure the CD86 and CD54 as the markers on cell surface with the flow cytometer. Depending on how much the markers have been increased after the treatment, if the increase is more than 150% in CD86 marker, you call it a sensitizer. If the increase is more than 200% in the CD54, we call it as also as a sensitizer. So this is the short explanation for this assay. Can you go to the next slide, please? Now, with these inputs, what you have to do is you have to score your results. This is nicely shown in this diagram. So if I have a HCLAT which is showing me the induction at a less concentration of than 10 micromolar, 10 microgram per ml, I will score it as 3 points. Respectively, at different concentration, whatever I see induction, I will give the scoring patterns for the HCLAT assay. In case of DPRA assay, depending on the lysine and cysteine depletion, the score will vary. So higher the depletion, higher the score, as shown over here. And then with the help of in silico approach, if the compound is predicted as a sensitizer, I will give a 0.1. If it is a non-sensitizer, I will give it a 0 point. In the end, I will sum up all the scores what I have got, which is shown in this table over there. If the score is in range of 6 and 7, we can easily classify that compound as 1A, which is a strong sensitizer. If it is score is in between 2 and 5, we can call it as moderate or a weak sensitizer. And if the score is less than those numbers, we call them as a non-sensitizers. So this is the classical score-based approach. With this, we can also predict the potency of the compound, which was lacking in 2x3 method. Can you go to the next slide, please? So with this, how good we are close to the nature or the how good we can predict the hazard or potency shown in this figure. So if with the classical approach, 2 out of 3, we could not able to get any potency out of it because this combination is not for that. But if you use the combination of version 1 and version 2, you get a very good hazard prediction as well as the potency, which is shown over here. And they are comparable with the LLNA predictions, which we use where we use animals as such. So these in vitro methods are in par with the approach what mentioned in this chart over there. Can you go to the next slide, please? So it's clear that whether you use these software-based direct one or whether you use freeware like OECD Toolbox, you get the similar output. With this basic introduction, I will go to small case studies in the next present. Can you go to the next slide, please? So for example, if you take a strong sensitizer like 2,4-dinitochlorobenzene, we know it reacts very rapidly with the cysteine, and you could see the up to 100% depletion when you incubate these things, this compound with the in the DPRA assay. And even lysine percentage is up to 30. So you have an average around 65. Since the average is greater than the 42%, we have to score it as 3. This is defined in this defined approach. If you look at the HCLAT results, the induction of the CD86 marker is at 2 microgram per ml level. So we have to score it as 3. Again, it's less than 10. So there will be a score of 3. And if I do a silico prediction, I can predict this compound as sensitizer. So if I add up the score of 3 plus 3 plus 1, the sum comes to 7. And with the 7, the potency score can be interpreted as the 1A classification, which is a strong sensitizer. So can you go to the next slide, please? So in the other example of 2-mercaptobenzothiazole, if one can see the cysteine depletion is up to 64%, we don't see any lysine depletion. The average is around 32%. It's a positive sensitizer, and this range of average range 32% is in between 22 and 42. So according to this defined approach, one has to give score of 2. If I look at the HCLAT results, the induction is occurred at 48 microgram per ml, so which is more than 10 microgram per ml and less than 150. The score would be again 2. And with the silico prediction, again, one can predict easily this is as the sensitizer. So this total score is 5. And since 5 is for the moderate or weak sensitizer, we can classify this compound as the what do you call moderate sensitizer. Can you go to the next example, please? So in this example, we have taken glycerol, and with glycerol, you don't see any cysteine depletion or lysine depletion. So this average is less than less than 6.38. So it's a non-sensitizer for the DPRA assay, and the score would be 0. If you look at the HCLAT results, and even at concentration of 5,000 microgram per ml, we don't see any induction in both the markers CD56 or CD86, and that's why again the score would be 0. In the silico prediction also, this comes as the non-sensitizers. So again, the total score is 0, and we can classify this compound easily as the non-sensitizers. Can you go to the next slide, please? So when it comes to pesticides, we know these are the chemicals which are used to control pests, insects, weeds, or the fungus, or insects, herbicide, or rodenticide. And generally they are absorbed to the skin, lungs, or digestive system, where your skin is the prime organ which comes into direct contact with these pesticides. And there are a lot of examples where the itchiness or redness of a skin has been observed. And many of these pesticides are also known to be allergen. And the question was, can we use them in this new paradigm? So can you go to the next slide, please? So this is one example of a model pesticide called as permethrin. Here you can see this pesticide reacts with the cysteine as well as the lysine residues, causing up to average of 23% depletion. And so we have to give the score as 2. If you look at the HCLAT assay, the concentration at which the 86 is expressed, we can get EC 1.5 at 45 microgram per ml. Same was the true for the EC 200 value. So the HCLAT scoring would also lead to a score of 2. In silico prediction, this comes as a sensitizer. So the average, if I sum up the scores, it comes to be 5, and it can be classified easily as the moderate or the weak sensitizer. Can you go to the next slide, please? Again, similar example, again insecticide called as methoxychlor. Here it reacts strongly with the cysteine residues, and we could see a depletion of 18% with the lysine, causing average depletion in 50% in the DPRA assay. So that we can easily term this as the sensitizer, and the score would be 3. If I look at the HCLAT result, again the score would be again 3. With the silico prediction, I could easily predict this as the sensitizer. So again, I have to give 1 as a score, and altogether I can classify this one as these strong sensitizers. Can you go back to the next slide, please? So the message which I want to give is these in vitro methods which we can use can be also used in case of pesticide. They can be used as the alternative for testing skin sensitization paradigm, and there is a need to use this OECD 497 guideline where we have to use this defined approach where we have to take the combined output of different adverse pathway key events and make some prediction about the nature of this compound. And this 497 is the guideline now gives us the approach how to classify this compound, not for as a sensitizer, but also as the potency, which would be help in the hazard prediction. Can you go to the next slide, please? Of course, there are challenges like as far as possible, try to avoid the contact with this pesticide, wear the proper protective clothes, what you have. You follow these safety instructions very meticulously, what is mentioned in the information about usage of pesticide. Wherever it's possible, try to substitute with the lower one. We have to educate our people who use these sensitizers, if it causes the sensitization, what are the precautions they need to take, and we have to also monitor the surveillance of the workers, how do they work actually in the field. So can you go to the next slide, please? So these are the references one can go through. Basically the top ones are the OECD guidelines, where these different key events are explained very nicely, OECD 497, and there are some interesting links like Link or QSA Toolbox or Prediction One, where we can look at the some sort of a silico prediction as well as the better for the better understanding of the nature of that compound. Can you go to the next slide, please? So now it's clear we all have to work together, responsibility to avoid the sensitization effect, and we have to use the in vitro methods to protect the environment as well as the human health. Can you go to the next slide, please? So at JRF, we have been using them for more than a decade, where different alternatives for skin corrosion, skin sensitization have been used over here. We also worked on novel phenomena where there was a limitation of the skin API assay, where one could not see the photo effect of UV rays on this compound. So that photo oxidation or photosensitization assay has been also developed at our end. The other assays for the eye irritation are also done, and we have also done some sort of the assay validation for dermal adsorption, where you could also measure how much is the compound penetrates through your skin, and then to cause any what you call further reactions. We also started different in silico approaches, which are mentioned over here. Can you go to the next slide, please? So these are the assays which we can give as a service. Yeah. Can you go to the next slide, please? And I thank you for your patient listening. Any questions related to these assays, we will be happy to answer.
Thank you very much, Dr. Date, for a very, informative presentation, and also a very much. We're very grateful to Dr. Hirna Pandya also for her presentation on skin sensitization. Thank you very much, ladies and gentlemen. My name is Vipin Seni. I'm the Chief Executive Officer of Biological Agri Solutions Association of India, commonly known as PSI. We have a couple of questions, and like rightly said, Dr. Date is all geared up to get along with it. So, but Dr. Date, I believe we'll go for ladies first in this order. So, Dr. Pandya, thank you very much for your insightful information on skin sensitization. Doctor, my question to you is in two folds. Can you expand a little bit more on the OECD ITA document 255 and 256? And also the next part will be that if you could underline the circumstances under which such NAM studies can, trigger an animal testing. So over to you, ma'am.
255 and 256 basically explains about the integrated approach and the tools which can be used. So if I understand the question correctly, that applicability domain is most important before starting the test, and existing information through in silico tools freely available should be first collected, and then going ahead, if the applicability domain makes a failure in the in vitro method, then the last resort which stays back is only the in vivo testing. But we do not encourage every time to do an in vivo testing first, but we would always want that the in vitro methods first should be tried. And if the classification category is not achieved, then you should go ahead only at the last resort for in vivo testing. So I hope that helps.
Yeah, that is, thank you. I believe it clarifies a little bit of it. I mean, the subject being new, many people will be having more questions on that regard. Probably will come to them later on. Dr. Date, my question to you is now directed that do these methods work on formulations also, or they are only on technical, or they work on both?
Very good question, sir. Basically, these are meant for the true technical compound, but there have been now attempts where one can use the formulations along with the true compound and try to get meaningful out of it. So, for example, there are some papers which talks about the different formulations are present and how the mixtures, the true compound along with the particular formulants, what would it lead to? So, but I have to agree, we have to come up with more examples to test the formulations as well as the true compound. How do they react and how to make meaningful out of those combinations.
Okay, thank you, Dr. Date. So, just in a word for a layman like me, does this work for formulations? Yes or no?
Yes.
Okay, fair enough. So that makes things more clearer to guys like me who understand it very less in this regard. I am seeing that we have Dr. S.K. Jain, the regulatory toxicologist expert from Central Insecticide Board and Registration Committee. Dr. Jain, are you there? I saw
Yes,
him.
yes.
Yeah, so Dr. Saab, I hope you were fairly impressed by the presentations given across by Dr. Pandya and Dr. Date. Sir, in this regard, as a regulator, would you like to, pose in a couple of questions just to throw about for the benefit of the audience on how these kind of approaches can benefit Indian pesticide registration in this regard?
Both the speakers, they have dealt the presentation nicely, but there are many queries regarding use of in vitro methods, in silico methods, and I personally feel as they are helpful for technical compounds, whether they are having any skin sensitization activity or not. But in many cases we find that technical doesn't have any problem with the skin sensitization, but formulations, they are more toxic, and we find there are more adverse effects with the formulation. So, but he agreed. Dr. Date said that we do with the technicals, but we have some issues with the formulations and they can work. But I don't think that at this stage we can go straight away for testing of formulations for skin sensitization using in vitro methods. They can be guiding tools for in vitro methods. If we find, based on the endpoints, they can have an indication. And if that is, as he suggested in one or two cases, that the value was 4 or 6, both cases, cysteine, lysine, and the average was 5 or 6, that case the compound is naturally non-sensitizer, and in that case, even if we go for animal testing, it would be non-sensitizer. So in that case, we need not to go for further animal testing, and that will be sufficient for accepting the data of in vitro studies along with in silico studies. So I agree that it is in nascent stage and early stage, but hopeful by advancement they will be helpful further in future. But we are trying to accept the non-animal methods. How far it can be useful and successful, particularly in pesticide regulatory system. But they have explained nicely, and I hope that it can be tried. All things cannot be accepted at this stage, all in vitro methods, but based on the results of the preliminary studies by using in vitro system could be helpful for doing in vitro studies and providing some data for not going for the animal studies.
Right. Thank you, Dr. Jan, for that very insightful thought upon it. And yeah, we need some little more time, I believe. Sir, just one question from you, Dr. Jan. Is it necessary that data to be available in vivo and then only an alternative NAM methodology can be utilized and taken across for further endpoints, or can NAMs also be done for where in vivo or animal studies have not been at all done in the country? Suppose in the case of new chemicals coming across, wherein probably there is no animal data involved. In that case, will such studies be, kind of considered?
In the first stage, we can go for NAMS studies using all the non-animal methods, and if the results indicate that this chemical, where we don't have any in vivo tox data available, and if that suggests that the endpoints they are quite stable and indicates to be non-toxic, then I think even if we go confirm by using the animal methods, the results must be nearly similar.
Okay, thank you, Dr. Jan. I mean, that's really something delightful to hear, and I believe registrants for pesticides in the country, many applicants will be, looking forward to such things if applicable. Dr. Pandya, yeah, Dr. Jan, you were saying something?
That's how we are trying, that we have to go for the alternatives, and in case-to-case basis, that every chemical cannot be accepted based on all in vitro methods or purely data available based on other studies other than animal studies. But in some cases, if the chemicals are found to be non-toxic based on these approaches, non-animal methods, then that could be accepted, and that we are trying to.
Right, sir. Right. I believe we lost Dr. Jan, but I believe we got the message what he wanted to convey across. Dr. Pandya, I have a question for you. You mentioned earlier in your presentation that nearly 50% of the industrial chemicals, skin sensitization is required to be done. I mean, they require skin sensitization toxicological evaluations. But then when we talk about non-animal approaches, testing approaches, we normally talk about only skin sensitization. I remember in 2015, prior to 2015, there was this talk going across one generation extended reproductive studies, which was slightly about to be, I'm not sure whether it was adapted by the EU in such, but it was still being validated at that point of time and was not acceptable in India also. That I'm not sure what the position is right now. But what I'm trying to ask you, that why do we normally hear about alternative methodologies only for skin sensitization, and we rarely hear about it in other toxicological evaluations? I mean, both Dr. Pandya and Dr. Date, you may also pitch in later on.
Yeah. So I would like to, you're very right, sir, that we always hear about the in vitro approach first and then doing the in vivo studies, because there are two sets of chemicals existing in around our arena. One are the data-rich substances and some are the data-poor substances. When we talk about data-rich substances, then we have a lot of options. There is an existing information which can be used and it can be coupled with these NAM methods, and you can bring about the skin sensitization prediction, and you can evaluate or come back with the answer on the endpoint, which is very important from the human health hazard assessment. While when we have a data poor substance, then you will have to really evaluate it more into the detail with all the methods which are available. And currently some of the regulatory bodies have, because the OECD has published this guidelines and documents in some of the regulatory arena, it is very strict that they would first ask for an in vitro approach. And if you do not derive, as I said, the classification category, and you do not have any information about that chemical, then they would allow you to go ahead for the further testing in the in vivo. But again, in vivo or vertebrate testing in some arenas, you have to take a strict permission from the authorities before doing those kind of tests. So as of now, in some countries, it is very highly regulated, and in some places, you have both the options available in vivo as well as in vitro for the testing.
Okay, thank you, Dr. Pandya. Dr. Date, your take on it.
So basically, why it is more talked about, because it's the first best example where the in vitro methods have taken a good role in ruling out. For example, if you look at cosmetics, we have 100% replaced the in vivo experiments, and we have shown that in vitro methods are capable of doing that. So being a first baby, everyone loves it, and there is lot of efforts put on the developing these guidelines. So if you look at the assays like DPRA and all, people have started working them from 2005. So it's a long hard work of many people coming together for 30 years to make some meaningful combination. And probably what we see is the output of it, which is gets highlighted most of the time, because like this we can, with the what do you call blessings from regulators, we would able to take some more challenges and try to imply in different conditions. So that's probably reason what I feel why it's more talked about.
Okay, thank you, Dr. Date. Continuing with you, Dr. Sam. Earlier in your presentation, you mentioned that these kind of in vitro studies tend to save time and cost. Time I can agree upon. Cost, sir, I would like to have a further clarification from you, because like you also mentioned in your presentation that if you do a single assay, you get around about a chance of 80 to 85%, getting it right. But then if you have a combination of assays, 2-3 combinations, and then probably you have a chance of 90-95% for validating, getting an accurate evaluation of the study. So, I mean, is it and it's a common rumor across the industry that alternative methodologies are far more costlier than animal studies. So, sir, is it really true, or I mean, what would you? I mean, I'll just like to have your opinion on this.
You want to go first or shall I go?
Yeah, I'll add something over here that Dr. Sani, you rightly said that time, of course, it is efficient for time. Cost, if we look around, everyone is concerned about the cost. There are a lot of efforts going on in the industry right now to bring down the cost for these kind of studies, and moreover, a lot of CROs are working around. And as Rahul said, that long history is associated with working on these chemicals, and a lot of historical databases have now been collected for which the chemicals are available, which are tested, and they will today or tomorrow, in some period of time, definitely this cost will also come down. if more and more acceptance is there in the industry and in by the regulators and the effort goes on, then definitely the cost of these assays will also come down. As of now, we do agree that some of the cell systems or the cell lines or the type of the softwares which are available, some are not freely available, they are very costly. But as and when the acceptance will increase in industry, the cost would come down. Rahul?
Yeah, I agree with you. For example, if you look at the acute toxicity, there's a guideline called as 129, where you can use simple fibroblast cells and look at the toxicity of those compounds on those cells. And once you get the number of LD50 predicted from this cell line experiment, you can reduce animal uses up to 25%. So a simple in vitro assay should not cost you much. And if you save 25% animals use for any acute oral toxicity test, that's here the I would say in vitro is cheaper than the animal experiments what you would get. So in respect to the skin sensitization, basically if you know the history, it's more used for the skin cosmetics industries. So there people don't want to take any chance that if you have a failure, it will result into a large what we call loss in terms of the hazard as well as for the public health. So people were more conscious, so more efforts were done to make it perfect. So probably that's the reason why we have some sort of a slightly bit higher cost to as per se to see. But in future, I believe these would reduce by a large extent. Not only for this one, for the other in vitro alternatives also.
Yeah, that's I'll take your word for that, both of you. That let's hope that it comes out to be cheaper in this regard. Well, coming back once again to the earlier question, which I felt was unanswered. See, there were two aspects. One is that there are limited norms in place at this point of time. And how long do you think that it's going to take the scientific community, the toxicological community, or the CROs to come across validated methods that can be read across or ready to replace the existing array, the whole list of exhaustive list of toxicological parameters to be done? And Dr. Pandya, for you, it is a specific note that I believe I was not clear whether the single generation or one generation reproductive study, extended study, is still has been validated by anybody apart from OECD or anything else, whether it's still acceptable. I'm not sure whether it's acceptable in India. I'm sure it isn't, but I may be wrong.
Yeah, I mean, in India, of course, people are not 100% going for it. But in outside other countries, it is now slightly more prevalent. Again, the endocrine toxicity area is one of the new emerging area where it has been putting a lot of weightage to have all the endpoints covered on the all different type of modalities. So extended one generation study are in existence and they are being requested by regulators, but again it's a very expensive and a heavily using number of animals, and there are a lot of efforts going on in other crop life groups how to bring down this method, and prior to that there are some in vitro studies which will be becoming the trigger point without which you cannot go ahead for direct in vivo testing. So again, there are a lot of OECD conceptual framework. If you will see, there are level 1, level 2, level 3, level 4, and level 5 studies in which these in vitro methods are there, and then you have to go for the higher tier or let's say extended one generation study. So in OECD conceptual framework, it is explained, but yes, it is not prevalently performed till now until unless there is a requirement from a regulator or a strict basis.
So cutting you right there short, shorten over there. See, I mean, why I've asked this particular question is that it is intended to my earlier question, how long it's going to take, because having been an executive director of Crop Life India also, wherein we took up this case during the finalization of the 2017 document, which was done, that point of time, 2015, and now we are in 2023, 8 years have gone by, and yet we are not there. So I mean, what do you see the time frame of where we can actually have probably 60 to 70% of NAM methodologies coming into place rather than going into for in vivo studies in this regard?
So very nice questions here. I would say in next 10 years, probably the situation would be different, where we would have a lot of those assays move to in vitro paradigm. I mean, few years back, 5 years back, probably very few of us knew what are the alternatives in cosmetics. And now it seems that every CRO or any, even in university level, we have raised the awareness where people are aware, okay, there is alternatives for skin sensitization, and these tests can be tried. So with the regulators' pressure as well as by the N2 of the scientific community, I guess in next 10 years situation would be much better, where most of those assays would be replaced by in vitro.
Okay, yeah. Thank you, sir. Thanks. And one thing I'd like to congratulate alternative skin sensitization methods in NAMs coming across that, like you very rightly said, I believe the world over now, cosmetics do not require skin sensitization studies to be done in vivo, and which is a remarkable feat in its own manner. A couple of years, I mean, it's been a decade now, probably, when this REACH legislation cropped in, and global suppliers, manufacturers, and all were asked to pool in resources to generate data and submit data on that. I'm not very sure, but I would be really interested to know whether in the REACH legislation also many industrial chemicals, obviously to the tune of a couple of 100,000. Were there any NAM skin sensitization studies accepted under the REACH legislation that can be taken up as an examples? I mean, any known, if you're aware of those things, that is.
I do not remember, Doctor, correctly on top of my head, but then they do have an acceptance level for the in vitro, and they are accepting these studies. That's what I understand. Rahul, would you like to add?
Yes, as you said, for the registration, there were a lot of chemicals to be registered in the limited span. So in vivo experiment with was not possible to do with the with such a big list. So they have started accepting the what you call in vitro results for sensitization as well as wherever possible. We have done a couple of studies for different companies for the registrations. Yes.
So, I mean, coming again to the same subject, I mean, if you could add upon that, I mean, do we have a list of those chemicals? Because if we have a list of those chemicals in which in vitro this skin sensitization methodical studies were acceptable and taken across, then probably if we look into the chemical structure of those chemicals in which that was acceptable, probably it can be extrapolated further across to other chemicals with similar chemical structures. I mean, is there any, I mean, thought in on this regard? Can it be explored, or whether the EU REACH legislation actually wanted to do something, or the CROs have tried to take it through that angle on skin sensitization so as to cater to a number of other similar chemical structure compounds?
From the CRO perspective, we are limited or the which compounds we have tested, we cannot disclose because of the confidentiality agreement between the tester and the labs. But I get your point.
I agree. I'm not talking about the CRO confidential. And what I'm talking about under the REACH legislation, since now it is being acceptable, these NAM procedures are being acceptable for skin sensitization, whether the EU or the OECD or ESHA have actually tried to document and to see whether they can be extrapolated to other similarly chemicals out over there. At least they can do some kind of peer review across on that regard.
Yes, a very good point, sir. I am not aware of these efforts, but if I know, I will definitely look into those things.
Okay.
Just to add, Mr. Seni,
yes, yes.
I mean, I have been in discussions with a lot of industry people, and I've been told that read across is one of the most commonest way of submitting MAM data, and the regulators have been accepting, even in India, information from read across, where you're extrapolating information from like a known chemical to an unknown chemical. So that's been happening. I mean, we may not be able to, give examples right now, but we know that this approach is being widely accepted.
No, that read across is in regard to similar, other issues in Vivo where we are trying to do those things. There may be certain things happening. What I really wanted to understand, like we have got peer-reviewed documents by EPA and others, right, which are acceptable by regulators the globe over. So the total concept of that thing. So when we have such kind of databases based on in vivo studies, then is any government or any regulator like the EPA or the OECD or Asia with those kind of resources trying to compile them into a review kind of document wherein that document can be taken across like a Bible for catering to a far more number of similar chemical structural compounds in that regard? That was what I'm trying to ask. Is anywhere, are you knowing whether something is happening across in any government or any kind of regulatory authority or so?
Dr. Seni, I'm not 100% sure, but I have went through one of the websites on the ECA where they have maintained a big database of these kind of chemicals, and they have put up the review reports, just like how you say a retrospective analysis, where they have seen around the chemicals, they have done the read across, they have done a lot of grouping of chemicals together, uh, looking at the structures and getting the data. So there are reports available under the ECA website which can be referred, but they will not be for a very exhaustive list. But of course, they are good reference documents, because ECA is a very big reviewing authority, and they review a lot of chemicals.
Fair enough, Dr. Pandya. Thank you very much for that thing. Yeah, Dr. Dutt.
I think there is very limited information available regarding, on this aspect, and we don't find much. Even we have tried to find out in different links and websites, but very limited information is available.
Right, Dr. Sub. Right.
Probably it just mentioned. Sorry. Probably it just mentioned the output of the test, whether it was validated in vivo or vitro. It won't give you more information, but it will give the final prediction. For example, particular compound is skin sensitive tested by in vitro method, but it won't give you more detail about it. I think you're right. If such information is more publicly available, it will make more sense to the people who want to extrapolate those things.
Yeah, because it was very. I was very curious. Like when we had more than 100,000 chemicals under the REACH legislation, a couple of 100,000 chemicals going across. Having new chemical compounds invented are more or less difficult, and they're all just a cross sectional of the existing ones. So probably we could come across some kind of document like that, like. Just giving you a vague example, like we've got the pesticide manual, we've got all the data out over there for a referral purpose, right? And to go even further ahead, we've got the Merck index, right, where covers a total huge list of chemicals coming across, and I can have all from the physical parameters to the toxicology to the ecotoxicology and all that kind of stuff. So that was one field which I believe can be done, and unfortunately in India still we are in a draft stage with the chemical safety and management rules, which is a kind of a replacement of the REACH legislation of the EU in our own sense out over here. So that is still pending across. Dr. Ankita knows more about it. She's been quite active across it, and Recently in biologicals, Dr. Pandya and Dr. Date, this question again comes to you. Dr. Jain, please feel free in joining in this regard. So how does this skin sensitization methodology help out or can help out in evaluation of biological agri inputs in this regard, maybe biofertilizer, microbial, botanical, a biopesticide or a biostimulant to that regard. So the skin sensitization is already there, I mean dermal toxicology is there and all. So how do you see or foresee whether it is really being acceptable, or the challenges vis-a-vis a chemical compound to a biological compound in this regard?
We are not sure whether can be accepted or not, or whether we have valid methods or not. It will depend what results we get from these methods for biologicals. So we have to see.
Right. Dr. Pandya, over to you.
Yeah, for biologicals, we have seen that not all of the biologicals would be able to 100% fall into the domain of testing of these chemicals. If I talk specifically, I remember one of the example that is a pheromone. It's a kind of an hormone which is used for the application, and that may not be 100% testable in the in vitro NAMs which are right now available. So we do see that some of the methods are applicable, but sometimes some of the methods may not also be applicable. So it is still an area where everyone is trying to understand that which would be the best fit for testing, the strategy kind of thing for biologicals.
Just continuing on this thing with you, Dr. Pandya, I mean, what kind of categorical biologicals do you believe that can be acceptable for NAM skin sensitizing tests? Can they be botanical in nature, or microbial, or like you rightly said, PGRs is a challenge, right? But in case of botanicals or microbials, is there anything acceptable there, or there are challenges there too in this regard? Or you may have observed that we can fairly go along confidently on either of these categories.
For botanicals, of course, there wouldn't be that much of a challenge. But for the others, I personally do not have any experience. Probably, if at the CRO level, if they have any experience, probably Rahul can share. But for botanics, yeah, it's acceptable.
Okay, fair enough. Right. Dr. Dutt.
As the earlier speakers have said, I mean, it varies from case to case. I mean, as long as you have a biological compound fits to the limitations or the criteria defined by the guidelines, for example, whether it's soluble, whether it has particular properties or not, one can test them. It becomes challenging if you have some sort of a complex molecules, mixtures. So these guidelines won't fit, or they clearly state that this is not applicable. So that would be a challenge in certain cases, whereas for certain cases where it's a straightforward, it should be not a big issue.
Right. Thank you. Thanks a lot. Dr. Ankita, you have any questions on this regard? I mean, anything else on skin sensitization, or I believe we have a couple of minutes more to spare. Otherwise, we can just thank our lovely speakers out over here for such an interesting interaction. Dr. Ankita? Okay. So Dr. Pandya and Dr. Date, thank you very much, and a special thanks to Dr. S.K. Jain also to having been joining us and giving us some kind of insight in this. Just one more question before we leave. We still have covered. You mentioned about endocrine disruptors. So, I mean, can the skin sensitization also help out in some kind of that kind of evaluation, or we need to see some other kind of thing going across?
I think so.
It's a very vague question, very layman's question, I should say.
We need to look into other aspects also. I mean, it's
okay.
Angle. Yeah.
Yeah.
I don't think skin sensitization will help you. I don't think so that only the results of skin sensitization study will help not to go for endocrine disruptor studies and all that.
Dr. Saab, since you're on the line, just one thing. We have got this dermal toxicity skin sensitization study, and then subdermal toxicities and all. So do we foresee that these NAM skin sensitizations can actually replace all these kind of acute the subchronic or chronic studies to deal with sensitization or the dermal aspects?
I don't think so. In coming time, it may take quite long time, as Dr. Date said, maybe 10 years or more. So it depends how the things go on for all these alternative methods.
Right, right, sir. Thank you. Dr. Ankita Pandey, would you like to, do you have any questions that you would like to, pose across? I believe we've lost her again due to some technical issues. Yeah. Yeah, she's, if anybody's seen that Hollywood movie Frozen, so she's actually looking like just like that. So, okay, that's on a lighter note. So yes, on behalf of PETA and the other organizations and the PSI, I thank both of you, Dr. Hirna Pandya and Dr. Rahul Date, for your wonderful, insightful presentation, your interaction, and deeply indebted for you to sharing that kind of knowledge with all of us. And I'm sure all the viewers who had joined us and who will view these recordings later on will really benefit from all of this. So thank you very much, all of you, for joining in. This is Vipin Sani signing off on behalf of the organizers and this webinar. Thank you very much, everybody. Thank you.
Thank you.
Thank you.
Thank you all.
Thank you, everybody.